I'm going to start this talk by taking us back in time by about 350 years or so. So the year is now 1675. We are in a Dutch town called Delft, and a man named Antony van Leeuwenhoek is about to change the world. This man is not a scientist, he is not a scholar or an academic of any description. He is a draper and a civic servant. But he has two very important qualities for our story today. He is insatiably curious about the world around him, and he is also, for his time, the maker of the world's most powerful microscopes. He makes them himself. He grinds glass into these impossibly perfect, tiny spherical lenses, which he then sandwiches between two brass plates to create an object that looks like this, kind of like a glorified door hinge. In this picture of that microscope, it is back to front and upside down, but never mind. This little object allows Van Leeuwenhoek to magnify things more effectively than anything else around at the time. And he turns it onto everything else around at the time, onto bits of animals and plants, and most notably onto drops of water, water that he collects from the canals and lakes of Delft, water that has accumulated in urns and vases outside his house. And wherever he looks, he always sees the same thing. He sees tiny little organisms way below the threshold of human perception, and in untold quantities. They are there, they are everywhere. They are moving about, clearly animated, clearly alive. And these are microbes, bacteria, and Leeuwenhoek has just become the first person in the history of the entire world to understand that these things exist, which to me makes this one of the most significant moments in the history of biology. This idea that below the threshold of our perception, there is a hidden world, and it is vast and complex. Since Leeuwenhoek's initial discovery, we have come to think of the microbial world as a source of death and decay. And there's good reason for that. In the decades after his observations, scientists very quickly recognized that many of the most infamous diseases to affect humanity in our history were the work of these so-called germs, everything from leprosy to tuberculosis, cholera, syphilis, gonorrhea, plague. All of these really nasty illnesses turned out to be the work of bacteria. And so we treated them as villains, as antagonists that we need to destroy unless they destroy us first. And this view of the microbial world still persists today. Disgust and fear are the primary emotions that come to mind when we think about microbes. If we look at headlines, they look like this. They equate to the presence of microbes on the surfaces around us with our failure to adequately cleanse those surfaces. I apologize to anyone here with a beard. I don't judge you. These headlines are deeply misleading and very unfair because only a few hundred kinds of bacteria are actually known to cause infectious diseases of the kind we talked about. The vast majority of them are either benign or indeed beneficial to us. And they are, in fact, the most dominant form of life on the planet. And they have been around for the longest time. So if we imagine that the entire 4.5 billion year history of our planet is represented by the space between this pillar and that one there, so that the Earth was created here 4.5 billion years ago, and that we are currently here in the present moment, then humans first evolved here. The dinosaurs reigned between around here and here. All the living things that you're familiar with, the animals and plants that you know and can see with your naked eyes, evolved around here. So for the rest of life's history, from the first moment when it arose, kind of around here, it was entirely microbial. You would not have been able to see it with your naked eyes, but that would have been fine because eyes only evolved around here. So The Earth is a microbial world. It shouldn't surprise us to find microbes on the surfaces around us, like this clicker or this laptop or this podium, because they are literally everywhere. And we animals, we humans, evolved in a planet that was already full of microbes, and we evolved to live with them and, in many cases, to depend upon them. Each of us today is a thriving community of bacteria and other microbes, a so-called microbiome. They live on our skin, in our guts, in our mouths, throughout our bodies. Many of you may have heard of this famous 10-to-1 ratio, this idea that we have 10 microbial cells in our bodies for each of our own human ones. This is a piece of fake news. It's the result of some back-of-the-envelope calculations using numbers that were pulled out of the air. In fact, the real ratio is more like 1-to-1. But that's still a lot of microbes in our bodies, tens of trillions, roughly one for each of our own human cells. So we are all just half the people who we think we are. The same is true if you look at every other animal that exists. This is a sponge. This is it's like the stuff that you get your bath sponges from. This is among the simplest of animals. It's just 2 layers of cells sandwiching a jelly-like filling. But sponges are also home to vast communities of microbes that do important things for them. Ants live in colonies that can number in their millions, but every ant is a colony of millions in itself. A polar bear that's walking through the Arctic tundra alone, seemingly isolated, is actually completely surrounded by and filled with life. Bar-headed geese fly microbes over the tops of the Himalayas. Sperm whales dive them down into the deepest oceans. And when these people first set foot on the moon, they were also taking a giant step for microbe kind. We are full of microbes. They go with us wherever we go. They also, really interestingly, help to build our bodies. So all of us develop from a single fertilized egg into an adult body full of trillions of cells. And that process of development doesn't unfold just under our own steam, according to the instructions encoded within our DNA. It also unfolds in partnership with and in conversation with the microbes that live within us. This is one of my favorite examples. A breastfeeding mother looks like she is nourishing her baby, and obviously she is. But around 10% of breast milk consists of these very complicated sugars called HMOs, or human milk oligosaccharides, which babies cannot digest. Those sugars are there to nourish microbes living inside the baby's gut. And in particular, certain strains and species, like the one on the slide, B. infantis, which have evolved the right genetic tools to break down those sugars with incredible efficiency. When they do, they release nutrients that then nourish the baby's gut cells, they seal up the lining of the gut, and they reduce inflammation in the gut. So a breastfeeding mother isn't just nourishing a baby, but she is nourishing an entire ecosystem within that baby. She is building an entire world. And like I said, that world builds us as well. We we rely upon the microbes that live in our bodies to shape and sculpt our organs. And their influence becomes very abundantly clear when you look at animals that lack microbes entirely. No such natural animals exist, but we can breed them. We can raise rats and mice inside these plastic sterile bubbles and disconnect them from the microbial world. So this rodent, which I'm holding in this picture, is an ecosystem of one. It does not contain multitudes. Now, the germaphobes in the room might think this is a pretty cushy existence, but in fact, this mouse has a host of problems. Its gut doesn't develop correctly. Its bones don't develop correctly. Its blood vessels, its brain, even its immune system develops improperly without that microbial exposure from a young age. And its multitude of woes attest to how important microbes are in shaping our bodies. So what about our brains? What about the organ that drives our behavior, from which we get our thoughts, our minds, our beliefs and ideas? Well, there's actually plenty of evidence from different parts of the natural world that microbes can also influence the way we think and the way we behave. A lot of the best examples come from the world of parasites. So this is a cricket, And coming out of that cricket is a horsehair worm. The horsehair worm infects crickets and addles their brains, making them inextricably drawn towards sources of water. The crickets leap into the water, they drown, and the worm wriggles out to continue its life. This is a worm changing the mind and controlling the behavior of the insect that it inhabits. This is the rabies virus. Rabies famously causes its host to act more aggressively because the virus passes on in the bites of its host. This is Toxoplasma Gondii. This is one of my favorite parasites. This naturally infects cats, which are the only animal in which it can sexually reproduce and complete its life cycle. Toxoplasma often finds its way into rats and mice, and when it does that, again, changes the behavior of those rodents, so that instead of doing the very sensible thing of running away from the smell of cats, it is inextricably drawn towards the smell of cats. The cat eats the rodent, Toxo gets to complete its life cycle. There is some evidence, depending on who you ask, it's either strong or shaky, about whether Toxo can influence human behavior too. People typically are exposed not through cats, but through eating contaminated meat. And large numbers of people around the world carry Toxo in their brains. Some estimates suggest that even 1 in 3 of us might have latent Toxo infections. There has been a lot of work on whether this can change our behavior and our personalities. Some of it is a little weak. Some of the strongest stuff relates to Toxo and the risk of schizophrenia. So what I want to point out here is that Toxoplasma Gondii is a single-celled parasite. This thing has no brain. This thing doesn't even have a body. It is one cell, and yet it can control the behavior of a rat, a mammal, a thing composed of trillions of cells. The rabies virus doesn't have a cell at all. It's debatable whether this thing is even alive. So you don't need complexity. You don't need a brain to affect a brain. Even the simplest of organisms can do it. So what about the microbes within our microbiome? What about those, for example, that live not as parasites, but as normal, benign, perhaps beneficial parts of our gut and other organs? Well, there is some evidence that those might also influence our behavior. And some of the most tantalizing hints of this were seen a long time ago in 1822. When a man named Alexis St. Martin, a fur trader operating in the Great Lakes region, came into an accident. This is not Alexis St. Martin. This is Tom Hardy from the film The Revenant. But this is roughly what I picture when I think about this story. And I couldn't find a good photo of Alexis St. Martin. He got into an accident where he took a musket shot to his side. The shot punctured his muscles, cracked his ribs, and eventually punctured his stomach too. He was in pretty bad shape when the only surgeon around, a man named William Beaumont, found him. But miraculously, Beaumont managed to save the man's life. The only catch was that St. Martin's stomach had attached itself to his body wall, creating a permanent hole from the outside world into his digestive system, what Beaumont called an accidental orifice. That hole never closed. It was there for the rest of St. Martin's days. And given that he couldn't obviously do fur trapping anymore, Beaumont took him on as a kind of manservant slash handyman slash experimental guinea pig. He would look at how different things influence Martin's rate of digestion. Occasionally, he would drop bits of food into the hole leading into his stomach to see how that food would then be digested. Through this work, Beaumont became the father of gastric physiology. And among his experiments, he noticed that as St. Martin's mood changed, so did his digestion. When the man became angry or irritated, and it's hard to imagine that you wouldn't be angry and irritated with a surgeon continually dropping food into your stomach, his digestion changed. And this was one of the first hints of something that I think we all intuitively understand, that there is this connection between the gut and the brain, the so-called gut-brain axis. We know that when we are sad or angry, it changes our appetite. Likewise, we know that when we are hungry, it changes our mood. We talk about being hangry. This gut-brain axis, this connection between those two organ systems, has a microbial component to it as well. There are many studies showing that changes in the gut microbiome, in the makeup and the membership of the communities in our gut, can also influence behavior. We see this a lot in rodent studies. Likewise, we see that changes in behavior. So if you subject rats or mice to stressful conditions of all kinds, you see changes in the makeup and membership of microbes living inside them. So these two things are very interrelated. Now, a lot of these studies are correlative, but there are signs that the microbes living in our gut can influence our behavior, or at least those of rats and mice. Many of those studies came out in 2011, which proved to be a very pivotal year for this field, and one in which it started becoming quite well-known and fashionable. So I'll take you through a few of these. In this one, we see that those germ-free mice that I told you about before, the ones that lack microbes of their own, they behave differently to those that live their lives full of microbes. They are less anxious and more prone to taking risks. But if you raise them, if you add microbes back to them at an early age, you normalize their behavior. So again, the presence or absence of microbes affects how these rats behave. This study took a slightly different tack. This took two different strains of lab rats, which behave differently. One is more timid and anxious than the other. It then took the microbes that normally live in those rats and cross-transplanted them. So you put those from the timid rat into the less timid rat, and lo and behold, their personalities also changed to an extent. And finally, this paper was particularly interesting because rather than using germ-free rodents, it used normal, healthy ones. And it found that one particular strain of bacteria, a lactobacillus strain, when administered to these rodents, these normal, healthy rodents, again, changed their behavior. It made them less anxious, so the rodents were more able to explore open territories or venture forth into exposed arenas. They were also more resilient to stress. If you drop them in water, they would paddle for longer before giving up, compared to rodents that hadn't been given this particular bacterium. So again, even in a rat that already contains a mouse that contains a normal, healthy microbiome, you can see differences depending on what you add to them. How does this work? How can microbes in the gut affect the brain? Well, there are several possibilities. There is the vagus nerve, a long nerve that connects the two organs. It essentially acts as a phone line between the two, sending signals between gut and brain. We know that microbes living in the gut can produce neurotransmitters, chemicals that we typically think of as brain signaling chemicals like serotonin or dopamine. They can even influence the production of these chemicals by cells in the gut. The immune system is also important. Here is an immune cell with some staphylococcus bacteria around it. The immune system travels around the body. It can act as a conduit of signals between disparate organ systems. So since 2011, there have been some really interesting studies linking the microbiome and certain neurological conditions too. So I'm going to tell you a bit about those. There's a really interesting study in Parkinson's disease. This is based on a mouse model, so a strain of mice that has been engineered to be especially prone to Parkinson's. If you raise these mice without microbes of their own under germ-free conditions, they are less likely to develop some of the motor symptoms that we associate with Parkinson's disease. If you add those microbes back at an early age, those symptoms also reappear. If you treat the mice with antibiotics, the symptoms go away. If you add microbes from people with Parkinson's disease to these mice, they again develop some of these motor problems more frequently. So, again, some evidence here that the microbes living in the gut can influence the course and progression of this disease of the brain. Now, what is going on here? How does this work? Some preliminary work from this study which suggests a possible mechanism. So we know that gut microbes can break down some of the foods we eat to produce chemicals called short-chain fatty acids, or SCFAs. This study showed that SCFAs can influence microglia, a type of immune cell that acts on the brain, on the synaptic connections between our neurons, and that can therefore influence the course of a disease that affects the brain. Now, this is still very early days, but one thing I want to point out here is that it is very easy in the world of the microbiome to categorize things as good or bad, and that's very rarely a good idea. For example, we know that SCFAs are typically thought of as a good thing. They have anti-inflammatory properties. They might be part of the reason why fiber is so healthy for us, because gut bacteria break fiber down, produce these chemicals which reduce inflammation in different parts of the body. So it's not necessarily the case that SCFAs are a bad thing that we should try and avoid. And that kind of contextuality is really important when thinking about this field. We will get back to that later. I also want to tell you about some studies that involve autism, or at least mice that have symptoms reminiscent of autism and have been studied to model that condition. One group had some really interesting work where they added a microbe called Bacteroides fragilis, or B-Frag, to mice that model the symptoms of autism. And they found changes in these rodents' behavior. They, again, were less anxious. They showed fewer repetitive movements. They were harder to startle. And in many of these changes, the scientists saw potential for treating some of the symptoms associated with extreme cases of autism. Now, why is this the case? Well, the idea is that inflammation in the gut causes the tight seals between cells that line the gut to become looser, allowing microbes and the chemicals produced by those microbes to enter the bloodstream and travel to other organs like the brain. Chemicals such as this one called 4-EPS, which seem to be particularly correlated with the degree of symptoms seen in this study. But B-Frag, because it has anti-inflammatory properties, reduces this, stems the flow of chemicals from the gut microbes to other parts of the body, and therefore reduces those symptoms. Now, several caveats that are important here. Firstly, this is one particular strain of B-Frag. Others have pro-inflammatory properties and will probably do exactly the opposite. So even two strains of the same species of microbe can have radically different outcomes depending on what they do. And that, again, is important when thinking about just how complex this world is. But also, more importantly, mice are not people, and mice do not have autism. Autism is a human disorder that we define in our own particular way. And we might be able to see some of those symptoms in the behavior of a rodent, but it's not exactly the same. And it behooves us to be a little bit skeptical when looking at what the results of these rodent studies actually mean for us. And I'm a big advocate for skepticism when thinking about microbiome studies, especially because there is often not a lot of it. Here is a set of headlines dealing with this topic of the gut-brain axis, with this idea that microbes influence our behavior. And you can see that many of them very immediately jump to this causal explanation, that gut bacteria may be to blame for things like anxiety. And some of them ascribe the bacteria automatically with this massive role. So this one, Parkinson's disease, may be traced to gut bacteria, as if that was the sole cause of this condition above all other potential causes. I don't think that's likely to be true, and I'm going to give you six things that you should bear in mind when reading news stories or papers about this particular field. One is that most of these studies involve mice, and there is a long history of laboratory rats and mice showing interesting findings that then don't translate across to humans, because we are different species with different biologies. Many of these studies are very small. The study of the microbiome is still, in many ways, a young and developing field. So people do small-scale exploratory studies. That's fine, but it also means that often these results are not as statistically rigorous as you might like. There are also lots of inconsistencies here. Partly that might be to do with with the small size of a lot of these studies. But a lot of the work that I showed you varies from one study to another. In some cases, adding a particular microbe makes mice more anxious. In some cases, less anxious. Sometimes the vagus nerve is important. Sometimes it's not important. There are these discrepancies and not an entirely consistent picture beyond the broader theme that microbes in the gut might be able to influence behavior in some way. There's a peculiar trend with this particular part of the microbiome field where there seem to be almost 10 times as many review papers as actual primary literature, which always makes me wonder, what exactly are you all reviewing? We need to draw a very stark line between statistical significance in studies like this and actual significance. So by this I mean, you might be able to show that the addition of a particular microbe changes the behavior of mice in ways that are relevant to things like Parkinson or autism or anxiety. But to what extent does that apply to us in our daily lives? To what extent do the microbes in our gut influence our risk of these various conditions? And the answer to that is just we don't know yet. And there is a big gulf between demonstrating that the microbes have an effect and actually establishing how big that effect is. And finally, the microbiome is not mechanism. And by this I mean that when I do talks like this, people often ask, like, so do the microbes in our gut explain X, Y, Z, which might be like, my risk of Parkinson's, or the fact that I feel happier after a meal, or all this sort of stuff. And it doesn't by itself. It suggests another thing that might influence all of these goings-on in our heads. But if we accept it as explanation, we then need to work out how all the things around us influence the microbiome, and then how the microbiome influences our brain. The microbiome is not an answer in itself. It just opens up more questions that then need to be answered. To really get at these questions and to establish the microbiome as something that is not just an influence on our behavior, but an important one, we'll need to do human studies. What I really want to see are randomized clinical trials showing that changing the microbiome in people can influence their behavior in important ways. There are very, very, very few studies like that. And I'm going to talk about one in particular and dissect it in some detail because I think it is instructive for our discussion today. This was a study of people with irritable bowel syndrome. This was a randomized controlled trial. It took people with irritable bowel syndrome who have either anxiety or depression, split them into two randomly assigned groups, and gave half of them this probiotic, a strain of Bifidobacterium longum, and then looked at how their scores, their behavior changed. So, two groups, one that received B. longum and one that received a placebo. Here's what happened. In the B. longum group, 14 people out of 22 showed reductions in their depression scores. Great. So only 7 people out of the 22 in the placebo group did. So this looks, on the face of it, really promising. Twice the number in the B. longum group than the placebo group. We're off to the races. But two things. Firstly, study found no differences in the microbiome between these two groups. If we're suggesting that adding this probiotic is changing their microbiome and therefore changing their behavior, surely you should see differences in the microbiome. There were also no differences in things like neurotransmitter levels between those two groups. Again, a bit curious. Another group of researchers looked at their data and concluded that the outcome is very fragile. By that, they mean that it doesn't take a lot, it wouldn't take a lot of extra people in the placebo group to also show reductions in depression scores to completely negate the statistically significant difference between those two groups. How many would it take? 2. Now, you could very well argue, but they didn't have another 2. That is not what they in fact saw. But there is reason to think that maybe they could have. If you actually look at the number of people with depression in those 2 groups, they are different. They shouldn't be different because technically those people were randomized, but they are different nonetheless. So there were only about, 13 versus 13 in the intervention group and only 8 in the placebo group who actually had some symptoms of depression. So instead of the probiotic changing their behavior, what we could instead be seeing is a regression to the mean. So just because you have more people who, in the probiotic group, you see a stronger effect, or a floor effect where there's not actually that much room for change in the placebo group. This is the type of stuff I'm talking about when I call for skepticism in this field. There are a lot of these really interesting, compelling early studies, but we need to think cautiously. And we need to think cautiously, especially when it comes to our attempts to manipulate, to change the microbiome, which is still very, very early and a little stumbling. So I mentioned probiotics here. Probiotics are products that supposedly contain beneficial bacteria that are meant to do all kinds of good things for your body. They don't really work. Probiotics seem to be pretty decent for treating certain kinds of infectious diarrhea. But for all the other benefits that are heaped upon them, the evidence suggests that they are medically underwhelming. Why is that? It actually makes perfect sense. The strains of bacteria in these products were chosen for historical reasons, because they are easy to grow and manufacture, not because they are actually important for us, or indeed because they are well suited to life in the body. They are, in fact, delisters of the microbiome. They've also been grown in industrial cultures for a very long time, so they're essentially domesticated. And they are found in very, very small quantities, much, much lower than the amounts that actually live inside us. So I liken taking probiotics to taking a small handful of zoo-reared captive animals and releasing them into the jungle, hoping that they will thrive. And they don't. They die. And that's what the bacteria in these things do. You swallow them, they don't stick around, they don't maintain a foothold. They just die and disappear or pass through. So it's no wonder they don't really have any effect. But the concept of using probiotics, of swallowing beneficial bacteria, is technically a sound one. Maybe you just need to choose the right one. That was the concept behind this clinical trial in India. This isn't about behavior, but it is important to talk about. They specifically chose a strain of bacteria that is present in the guts of babies, and they gave it to certain babies along with a sugar, a food stuff that was meant to nourish that particular microbe. And as a result of that, they saw a 40% reduction in the rate of sepsis between the babies that received this combination of bacteria plus food versus those that did not. And this was a study of 4,500 babies, not 22. This is the type of evidence that I would like to see to establish that the microbiome really does have an influence on the brain. Now, maybe instead of just giving one particular microbe, we could give people entire communities of microbes, vast collections of them. This is certainly the logic behind a rather unorthodox treatment called the fecal transplant, which is exactly what it sounds like. You take stool and you give them to sick people. Actually, look, I've prepared this highly technical slide. It is very difficult to find a good image for a fecal transplant. So I have used emoji. You take a sick person and you give them stool from a healthy person and they get better. That's why they pay me the big bucks. think of it like this. It is like an ecosystem transplant. It's like returfing a lawn that has been overgrown with weeds with fresh green pasture. And in this analogy, the weed is typically a bacterium called Clostridium difficile, or C. diff, which causes severe, recurring, intractable cases of diarrhea. It's very hard to treat. Give people antibiotics, and you'll typically cure only about 25% of C. diff cases. Give them a fecal transplant, and you will cure upwards of 90%. And that's been established in a variety of clinical trials now. So, curiously, poo transplants seem to be the best way we have of treating infections with C. diff. The problem here is that C. diff is a very unusual case. Fecal transplants have been tried with a number of other disorders, with things like inflammatory bowel disease, irritable bowel syndrome, and many, many more. And in all of these cases, success rates are lower and much less consistent. Because even when we are taking a full community of microbes from one person and putting them in another, it is hard to make these things stick. It is hard to know what the outcome of that transplant will be in pretty much every situation except C. diff, which is a gut that has already been turned into like a monoculture of one particular nasty thing. And our inability to sort of predict how well fecal transplants work bears out in studies where scientists have tried to duplicate the effect of this treatment by giving people specific cocktails of microbes without the fecal element to it. Those synthetic pills have been tried in various clinical trials, and even the most promising of these recently flunked out of a phase 2, which did allow me to write this headline. You thought you were here to learn about the gut-brain axis. In fact, it's just dad jokes. Okay, so I tell you this because, again, I'm a big advocate for caution, and there is often not that much of it here, especially when it comes to fecal transplants, which are actually very, very easy to carry out. People can do them in the comfort of their own homes, using little more than a blender and some tubing and a funnel. And people have done this. Some have done it for good reason, because, as you might imagine, in the early days of this therapy, doctors weren't so keen on it, thought it was quite hokey, so people who had C. diff infections tried them out for themselves. But loads of people are now doing them, and rather worryingly, the scientists who work in this field have many reports of parents trying this on kids with autism. Now, why is that a bad idea beyond just the ick factor? Well, in those studies I told you about with the germ-free mice, there are many cases in which you move microbiomes from a sick animal into a germ-free healthy one, and the symptoms of those conditions transfer over, everything from obesity to colorectal cancer to heart disease and so on. So when fecal transplants are done, people screen for obvious pathogens, for things like HIV or hepatitis. Now, most donors are required to have a healthy weight, have no history of taking antibiotics, be basically paragons of human health. But that's not the case when we're thinking about DIY transplants. And even in hospital settings, it's quite difficult to really screen for all the potential outcomes of this. Now, if you are someone who's suffering from C. diff and you might die, maybe that's a risk worth taking. If you are a kid, I think the equation changes, especially when we've talked about how the microbiome can alter the way our bodies develop over time. This is a researcher named Emma Allen Vercoe who studies this. And I think her quote makes a lot of sense, that this is adult poo in a pediatric population. What if you're basically setting someone up for something as awful as colorectal cancer later on in life? Controlling the microbiome is not simple. It is not like the simple arithmetic that we are used to thinking of when it comes to medicine. If someone has a vitamin deficiency, you add the thing that they're missing back in, and they're better. If you have a viral infection, you subtract the nasty thing they have, and they're better. Controlling the microbiome is not that. It is like engineering an ecosystem. It is changing a community as complex as a coral reef or a grassland or a jungle with all the complexity that that entails. And it's not even clear to me or to some others in the field whether we have the basic conceptual foundations of this sorted out yet. By which I mean, the traditional approach to studying how disease is affected by the microbiome is to take sick people and healthy people, or sick mice and healthy rat mice, compare the microbes inside them, look for differences between the two, and then you assume that like any differences are related to their disease. If there's too much of a particular bacterium, maybe you can get rid of it and they'll feel better. If there's too little of another, you add that back in and maybe they'll feel better. So the idea that there might be predictable differences between healthy and sick populations is kind of like the cornerstone of much of the research in this field. This was a paper that came out last year that I really like, which calls that whole idea into question. It looked at a lot of evidence from coral reefs to people with cystic fibrosis to chimpanzees with HIV, and it found that in many cases, individuals that are stressed or sick don't have predictable changes in their microbiome. They just have very chaotic changes. It's not that their microbiomes shift in one particular direction, it's that they shift in all directions, that the host loses the ability to adequately control which species and strains live within it, and therefore everything just goes out of whack. They call this the Anna Karenina principle, on the grounds that every unhealthy microbiome is unhealthy in its own way. But if this is right, it poses an infinitely harder problem when we think about how to define whether a particular community's health is good for you or not, and then how to even go about changing that. So we're still at a very early stage of this field. I think it's fascinating. I think there are a few areas of biology that are more fascinating because we're learning that these organisms that were around way before us and that are everywhere have these really powerful influences on different aspects of our lives. But we don't still have a complete roster of which ones live in our bodies. We cannot explain the vast majority of the variation between us. So we all share 99-point-something% of our DNA, but our microbiomes are much, much more variable. And we can only explain maybe 10 to 20% of that variation at most. So what makes our microbiomes different? We largely don't know. And if we go out into the wider world, our ignorance becomes even deeper. So I'm going to end on this picture. This is a tree of life. This is a pictorial representation of everything that exists on the planet. Now, if I zoom into the bottom of this diagram, onto that little green twig at the very edge of the image, that is you. That is you and every other animal, plant, every other living thing that you are typically familiar with, concentrated in that one tiny twig. The rest of this tree is entirely microbial. And that purple spray to the right, that would not have been on this image if I'd shown it to you 5 years ago. So imagine what this tree would look like 5 years from now, 10 years from now, 100 years from now. The microbial world forms the vast majority of life on Earth, and we are only scratching its surface. And that's what I want to leave you with if you're interested in this topic, if you're interested in my book. We still have a lot to learn, but it's worth making the journey because these are things that live in us, that live on us, they shape us, they influence us, and we don't really truly understand ourselves if we do not understand our relationships with them. Thank you very much.